Welcome back to The Explainer. Today, we're doing something really cool. We are decoding the microscopic battles and, well, cellular negotiations that happen the exact second you swallow a medication. It is a high-stakes, completely invisible world in there, so let's dive right in. Here is our roadmap for today. First, what is pharmacodynamics? 2, cellular locks and keys. 3, agonists versus antagonists. 4, measuring a drug's effect. And finally, number 5, the safe therapeutic window.
All right, kicking things off with Section 1, what is pharmacodynamics, the body versus the drug?
So, pharmacodynamics is formally defined as the study of the biochemical and physiologic effects of drugs and their mechanisms of action. But honestly, simply put, it just means what the drug does to the body. Now, we gotta contrast this for a second with its exact opposite, which is pharmacokinetics. And that's what your body does to the drug.
Now, what's really interesting here is how stark this difference actually is visually. On the left side, we've got pharmacodynamics. The drug is actively acting on the body's receptors. That's triggering a therapeutic effect, like lowering blood pressure or maybe a side effect like nausea. Then, over on the right, pharmacokinetics is the actual journey of the physical pill itself. It's how your body absorbs it into the bloodstream, distributes it, metabolizes it in the liver, and finally excretes it. But for this explainer, we are focusing entirely on the left side, the dynamic impact that the drug has on you.
Moving into Section 2, cellular locks and keys, or what I like to call the microscopic hubs. So, our bodies use these tiny signal detectors called receptors. Basically, you can think of these as cellular locks just waiting for a very specific key. And no, you absolutely cannot just pick them. There are 4 main types of locks on our cells: ligand-gated ion channels, G-protein-coupled receptors, enzyme-linked receptors, and intracellular receptors. When a drug binds to one of these, it forms a drug-receptor complex. And the absolute golden rule here? The magnitude of any cellular response you get is directly proportional to the number of those complexes that are formed.
And get this, the speed of your medication depends entirely on the type of lock it targets. Take a look at the far left. Ligand-gated ion channels react almost instantly. We're talking mere milliseconds. But if you glance all the way over to the right at the nuclear receptors, which, by the way, are completely inside the cell and deal with gene transcription, those can take hours or even days to actually produce an effect.
Which brings us to Section 3, agonists versus antagonists, the cast of characters. All right, let's meet our main players. A full agonist is a drug with an intrinsic activity of exactly 1. It perfectly mimics the body's natural molecules to produce a maximal biological response. Imagine a drug like phenylephrine sliding perfectly into the lock and turning it completely to open the door. Boom. An antagonist, on the other hand, has an intrinsic activity of absolutely 0. It binds super tightly, sure, but it doesn't activate anything. It acts exactly like a broken key jammed in your front door. It just completely blocks anything else from getting inside. But antagonists? They are incredibly clever, and they use a bunch of different strategies to block drug effects in your body. First up, you have competitive antagonists. They literally fight for the exact same binding site as the agonist. Then you've got non-competitive antagonists. These take it a whole step further by covalently binding to the receptor, basically locking it down permanently. Chemical antagonists take a totally different route. They use ionic binding to literally tie up the agonist right in the bloodstream so it can't interact at all. And finally, physiological antagonists. These guys actually bind to a completely separate receptor, but they kick off a totally opposite effect in the body just to cancel out that first drug. And then, right in the middle, we have the highly nuanced partial agonist. This is a drug with an intrinsic activity that's greater than 0, but still less than 1. See, even if a partial agonist binds to every single available receptor, it still can't produce that same maximal response as a full agonist. Think of it way more like a volume dial or a dimmer switch rather than just a simple on and off switch. Aripiprazole is actually a really brilliant example of this. It acts as a partial agonist on dopamine receptors, which means it can turn down overactive dopamine pathways while gently bumping up the underactive ones. All right, Section 4, measuring the drug's effect, or translating the math. So how do doctors actually translate all these microscopic interactions into clinical math? Well, we track this using dose-response curves, which really requires understanding 2 massive concepts, potency and efficacy. Potency is simply the amount or the physical dose of a drug you need to take to get a given effect.
Efficacy, on the flip side, is the maximal response that drug can possibly elicit once it actually binds to the receptors. So, stated super simply, potency is how much you need to take, and efficacy is how well it actually works.
Okay, let's dive into this and map it out by comparing 3 different drugs. If you look at drug A, that line on the far left, it achieves 50% of its biological effect at a much, much lower concentration than drug B. That means drug A is highly potent. You need very little of it to see results. But look over at drug C on the right. Notice how that line plateaus dramatically lower than the other two? Yeah, no matter how much of drug C you pump into the system, it is literally never going to reach the maximal effect of drugs A and B. It just has the lowest overall efficacy. Now, when tracking these responses, we categorize them in 2 ways. A graded dose response is when the effect is continuous and gradual, so it increases as the dose increases within just a single individual patient. But a quantal dose response? That is totally binary. It's an all or nothing effect. Sort of like asking, did their blood pressure drop past a specific threshold or not? And we observe that across a huge population just to see exactly how many people respond at all. Moving to our final part, Section 5, the safe therapeutic window and the very real stakes. This is where we talk about the high-stakes reality of pharmacology, which is that incredibly fine line between a cure and a poison. To actually find that line, we have to rely on population metrics using 3 crucial acronyms. First is ED50, the median effective dose. This is the exact dose where 50% of individuals show the desired therapeutic effect. Then we have TD50, the median toxic dose, which is where 50% of the population experiences a toxic side effect. And lastly, LD50, that's the median lethal dose, usually figured out in animal trials, representing the exact dose required to produce death in half the subjects. So the most crucial part here is how we mathematically calculate a drug's safety margin using those exact metrics. We do this using the therapeutic index, or TI. The formula is beautifully simple. It's just the median lethal dose divided by the median effective dose. By comparing the dose that causes harm to the dose that heals, we get a crystal clear number that represents the safety buffer of absolutely any medication. And this brilliantly illustrates exactly what we're talking about. That buffer zone sits perfectly wedged right between the therapeutic effect curve and the toxic effect curve. The actual distance between them, that's the therapeutic index. And the dosage range right between the minimum therapeutic dose and the minimum toxic dose is your therapeutic window. Now, if a drug has a wide window, it's generally very, very safe. But if it has a narrow therapeutic window, like the blood thinner warfarin, for example, that dosage has to be managed with absolute pinpoint precision to prevent toxicity. It really is an intricate, beautiful, and honestly, sometimes precarious microscopic dance happening inside your cells every single day. So the next time you take a common over-the-counter medication, I want you to consider this. How wide is its therapeutic window, and are you staying safely within it? Thank you so much for joining me on this explainer today, and definitely keep exploring the incredible science happening inside you. See you next time.